with me Dr. Fahim. So we both will chair this session. And mm, let me just to explain the program, how we will proceed. The first lecture will be by Dr. Zahir Ali. That's on number 3rd in that list. He will deliver uh, his talk online on Skype, and then we will follow the sequence. And so, Dr. Zahir Ali uh, will present his talk on a CRISPR-Cas9 based lateral flow assay for plant genotyping and phytopathogens diagnostics. Dr. Zahir Ali is a research scientist at King Abdullah University of Science and Technology in Jeddah, Saudi Arabia. So in just a few minutes, we will start.
Dr. Zahir, can you make it full screen, please?
So can we please ask him to start? Zahir Ali, I'm Dr. Nasir Saeed. I'm chairing this session.
Hi, Dr. Saeed.
How are you?
Yeah, fine. Can you see my screen now?
Yes.
Okay.
So can we start now?
Yep.
Yeah. Okay.
Yep. Hi, everyone. Good morning. I'm Zahir Ali, working as a research scientist at KAUST. Today, I will present our recent work about the point-of-care detection, the point-of-care diagnostic system for plant genotyping and pathogen diagnostics. And thanks for the committee members to invite me for this talk. So I will start why we need a CRISPR, or why we need a point-of-care diagnostic system. As we know that the agriculture system is totally dependent. The agriculture system is basically dependent on the pathogen diagnostic, and if we can diagnose the pathogens early, we can go for sustainable agriculture. This is one of the main goals, but unfortunately, early point-of-care detection or the point-of-care in field detections, these things are not available specifically for plants. So we think that if there is any system available that we can use for point-of-care or in field detection, it can help the farmers and it can help for sustainable agriculture. The second point that plant genotyping, it's very important in agriculture. And if we know a genotype is there or a sequence is there which is going for specific value in breeding, and we can incorporate it into the plants, or we can use it. Unfortunately, now, like compared to the other system, we have the CRISPR-Cas system where we can use very easily any, we can integrate, or we can remove, or we can change any sequence of interest for our specific purposes. So if we know the genotype early, or if we know a plant can contain a specific sequence, then this can help us to devise method for sustainable agriculture. But how we are doing the pathogen diagnostic and plant genotyping? We are using multiple approaches, like which include the field-based approaches to look for the resistance as well as for the better crops and for better varieties. Or we are using the lab-based approaches for pathogen diagnostic, where we are using mainly the PCR and RT-PCR and all these stuff. But if we are talking about the in-field, none of these can be used at the spot. Why? Because mainly the design is very difficult for all these, and it's like they need specialized trainings. The personnel should be specialized in specific trainings. And then these things take a lot of, they are costly. They take a lot of money if we are doing field trials and if we are doing the lab-based research for pathogen detection and plant genotyping. And one of the main problem, another problem is the test results. It's take from days to months, and sometimes even like we don't have that much time. Like specifically for pathogens, if we didn't succeed it in the first few days, then we cannot control a disease. So look, based on these things, we think that there should be a point of care diagnostic system for sustainable agriculture. But what it could be, a system that we can use in the field within no time for pathogen diagnostic, and if we need it for the plant genotyping, that will be great. But what it could be, and we think that the next generation point of care diagnostic with the CRISPR-Cas system can provide us a very easy system which we can use in the field for specific pathogen diagnostic as well as for genotyping of the plants. So based on this one, we think that the CRISPR-Cas system can be used. Why? Because as we all know now that actually the CRISPR-Cas system consists of a specific sequence that is present in the, that is inserted into the Cas array. And then this sequence, once we put any sequence like the natural system, like the bacterial natural system, they are using the immune recognition system from the virus genome. So, and this can be programmed. So we think that if we put specific sequences into this system, we can use this system, or we can exploit this system for any targeted genome modification as well as for diagnostic. And how in the natural system it's working, like it's just need just express this sequence combined with this protein, and then it can go to any target sequence where they can make a cut. This is for genome editing, but can we use this for targeted detection of the pathogen? And the answer is yes. Like instead of cutting, if we use a dead Cas9 and we can use the guide sequence, we can go to any target. So keeping this in mind, we think that like this system can be used. Why this system can be used? Because we need just a 20 nucleotide sequence from a genotype or from a pathogen, just preceding by the NGG sequence, which we normally call the PAM. And then if we express a guide sequence for this target, the Cas9 can go there. And if we put a reporter system on the Cas9, then we can look for this reporter system, and that can tell us the pathogen or the genotype is there or not. So basically, we are using the CRISPR-Cas system, but instead of the normal Cas system, we are using the DCas, which is the dead Cas9, because we don't want to cut the genome, but we want just to stick to a specific sequence for our reporter system. And based on these things, that we think that we can use this system with a lateral flow assay for detection of pathogens and genotyping. When we look into the normal lateral flow strips, that is like the normal type of the pregnancy strips and these normal available strips, basically they are composed of different parts. Like here we have the nanoparticles, which is bounded with the FAM antibody, and we have the streptavidin line, and then the anti-FAM antibody. So we think that we can take advantage of this system combined with the CRISPR-Cas system. If we can put, like if we have a DCas, which is dead Cas9, it can bind to a sequence by the guide sequence, by the guide RNAs. And if we can biotinylate it, so biotin, and this is well-known factor that biotin can interact with the streptavidin. So if our guide sequence is there, and if the target is there, it will bind with this one, the DCas9, and then this biotin can stick here. And then we can see a very clear system, and we can develop a very clear system for the field diagnostic. And basically, if we can do this, what we need in the field, we just need a simple heating system. We can use a heat block, or we can use a heat bath system. and our reagents, and the lateral flow strips, tubes, pipettes, and tips, and that's all. And here we don't need also a lot of equipments, a lot of different material. And with this simple system, we can complete the whole process within an hour for genotyping or for pathogen diagnostics. So to develop this system, we made a working principle for the bioscan, and we think that this system should work. Like, this is our target sequence, which we can amplify with a FAM oligo or primers. And if we can amplify this one, because the guide sequence is specifically designed for this one, so this can bring, like the guide sequence can bring this into the target. And then we have the nanoparticles anti-FAM antibody, and which can make this complex. So if we have this complex, now if we put this complex into the lateral flow strips, here we can see that there is no coloring or no strips development. But here, if we put the sample, and if this sample is composed of this complex, then definitely this material will bind with the streptavidin. And here, this we can see, like, if a positive result will look like this, that we have the test line developed, and this is just a control line. But if the result is negative, or the pathogen or the genotype is not there, we will not see any development of any color line in this position. So following this simple protocol and simple approach, we think that we can develop a system. So the first thing to develop this DCAS9, what we did a dual promoter, a dual plasmid system, where we put Cas9 with the AV tag. AV tag is specifically responsible for biotinylation. And then there is another protein which can do the biotinylation, which bring the biotin to the AV tag. That is BER-A. So here, what we did, we just put this under the T7 promoter with this one in bacterial system under the T7 promoter. This one will produce the BER-A, which will bring the biotin. This is the BER-A, and the biotin will be conjugated with the bio-DCAS9. So what we did, we expressed this system in the bacteria, and then we use, we express this, we purify it, and here we can see that we can purify the protein. But is this protein is biotinylated or not? We use the anti-biotin antibody, and here we can see very clearly that yes, our protein is, our purified protein is biotinylated. So then the next step, we also express the gRNA or the guide sequence for these things for detection of the targets or genotyping. And then we can apply it to our system that we collect the sample, just extract on the field. We can put it into the tube with reagents, and then we can proceed with the simple block. And after the reagents are done for 37 degrees C for 15 to 30 minutes, we can apply this into the lateral flow strips, and we can see our results. So to be 100% sure that yes, the system is working, first we confirm that that our system is working. We just take like two target sequences. We put the non-specific gRNA and the specific gRNA. Here in the specific gRNA system to the target, we can see that yes, this DNA is specifically recognized by our system. We repeat this with another target as well, and here we can confirm that target 1 as well as target 2, they are specifically detected. Then we confirm it with a system where we put like multiple negative and positive on our strips, that there is no cross-contamination, like no gRNA, like no protein, no DNA, no target or the wrong target or the wrong gRNA. And here we can see that our system is specifically detecting the material. But the next question is like, can we use this for the plant genotyping? Can we use this system for the plant genotyping and pathogen diagnostic? For that, we use first we select the two target sequences. Sorry, actually, I cannot see my mouse, so that's why I just. Can you see my screen? Hello?
Yes.
Yeah, sorry, I cannot see my mouse. That's why there was some problem. Yep. So as a first target, we select the LR34 in the wheat genome. As we know that the LR34 is exactly similar to the wild type, except 3 nucleotides or single amino acid is missing, which make this protein unsusceptible to the pathogen, or the pathway is blocked for the pathogen. So here, a specific target we select, and this target we keep it near to our PALM sequence. So if it is the wild type, the gRNA will not bind, because here we have extra nucleotide in the guide, and the Cas9 system cannot tolerate any non-specific nucleotide here. But our guide RNA was specifically designed for the LR34 genotype. And here we can see that our guide RNA, our Cas system can specifically bind with only the genotype that contain the LR34. To confirm this system that it's not binding with the wild type, but it's binding with the specific allele of LR34, we select 2 lines, which is the Keriga and the Avicet S. Keriga is the one line that already contain the LR34, and here we can see the bioscan can specifically detect the LR34. But Avicet don't contain, and it have the wild type sequence, and here we don't see any detection. And then we reconfirm that this system is working properly. But then the main question is like how we can use this in agriculture, or how we can use this in the breeding system. So we select the land races that are like the normal wheat available in the field or with the farmers, and we bring different land races, and we check that which land races, like for example, have the similar sequence as the Keriga. And this is a totally blind approach. And here we apply 14 different land races. And here we can see that one land races, like even maybe the farmer don't know, but this land race already contain the LR34 sequence. But to confirm that this is really the sequence and not any artifact of the system, we sequence this land race, the LR34 sequence. And here we can see that this sequence, this land race number 8, it specifically contains the similar sequence as the Keriga. And this confirmed that if we want to use this for the genotyping of the plants, we can use it. To reconfirm that this system can be used in the synthetic biology, or also it can be used in the genome editing, we use this system for our CRISPR-Cas9 created synthetic alleles. So we created actually specific alleles in plants that are resistant to herbicide, like SGR3 and SGR5. So we specifically created this in plants, but then how we can confirm? There are different methods of confirmation, but they are difficult, and we want a simple method for the detection. So here we can see that when we design a specific gRNA for our created genotype or synthetic allele, we can detect it through the lateral flow system with the our bioscan approach. So here we can see that both the rices that we created, we can see specifically when we are using specific gRNA against the SGR3 or against the SGR5. And compared to the wild type, where there is no modification or no synthetic allele, the system is not detecting any such kind of sequence. Then the next question is like, can we use it for other purposes? And one of the main problem is the confirmation of transgene. So normally transgene are confirmed by the PCR or by the RT-PCR, but can we detect transgene at point of care or in the field or in the seeds? And for that, as we know that normally for transgene integration, the tDNA system is used in plant, in the dicot as well as in the monocot. The only thing difference is like the 35S promoter or the promoter, ubiquitin promoter that are used. So we designed specifically our bioscan system for the dicot ubiquitin and for the monocot ubiquitin. And we confirm that this system can be detected, or these genes can be detected in for transgene or not. And here we can see that the bioscan can specifically detect both ubiquitin as well as 35S promoter in tobacco, as well as it can detect it in the Oryza sativa, the rice genome. So this confirmed that this system can be used for the transgene confirmation, and if we are trading something or if there is a trading limitation, we can confirm the seeds that these are transgenic or non-transgenic. So this is a proof of concept that yes, this system can be used in the field or even at the market, that the seed is transgenic or non-transgenic. Then the other main question is like, can we use this system for the specific detection of the phytopathogen, specifically at the early stage of infection? So for that, we select like the plant viruses, we select the plant fungi, and we also select like both of the viruses, DNA viruses as well as the RNA viruses. And we select two of the wheat pathogen, the Magnoforte and the Puccinia, and we select the two bacterial strains that are causing, they are pathogenic to the plants, like Pseudomonas syringae and Agrobacterium tumefaciens. And here we can see that our system can specifically confirm, like as a DNA virus representative, we use the T-wild-CV. As a RNA, we select the TMV and we select the PVY for specific detection. And here we can see that these viruses can be efficiently detected with the bioscan system. And then if we want to confirm that a plant contains multiple viruses, we can also do the same assay, like a duplex assay on the same sample. And here we can see that if like we co-infiltrate the two viruses, and here we can see that both viruses, they can replicate in the plant and we can detect both of these viruses. Similarly, we have like the wheat genomic DNA, which was infected with the Magnoforte. And here we can see specifically that we can detect the two specific sites of the Magnoforte genome by our bioscan assay. Here we can see that the Puccinia, when we have the plants, the wheat plants infected with the Puccinia, we can detect specifically the Puccinia pathogen with the system. And as a proof of concept, next we infect the plants with the Pseudomonas syringae, and we can detect these plants specifically with the bioscan system. And normally in the labs, we are using Agrobacterium tumefaciens. So we just infect some plants with tumefaciens, and then we select detect the Agrobacterium tumefaciens is present in the plants or not. So this confirms that yes, this system can be used for genotyping, for transgene confirmation, as well as for pathogen detection. Then the other question, or one of the big question, like the point of care diagnostic. What we means by point of care diagnostic is that like it should not require like the big equipments like -80, -20, and even 4 degrees C, because these things are not possible in the field. So a system, a point of care system should be that which don't require all these extra stuff. As we know that bioscan don't require any PCR or anything, but here we also want to remove this one. And also another one, like we need quick extraction. We don't need like centrifugation in the field and these things. So we devised specific quick extraction method, which can be used to extract the RNA or DNA for bioscan platform. And then to confirm that our system is stable in the field condition, we keep the bioscan reagent for like weeks. We have like first week, second week, third week, and fourth week in the room temperature, 4 degrees C and -20. And here we can see that at room temperature, the bioscan reagent can be stored or can be used till fourth week of the storage. So this confirms that this system can be used. It don't require any of these material or any of these equipments. And it can be stored at room temperature and can be used till like for a month. So this means that if we want to check our plants again and again in the field for a month, this system is available. And then I will come to the conclusion that the BioScan reagent can be produced easily in the lab. And BioScan is comparable in sensitivity with the commercial RT-PCR or the qPCR. And BioScan doesn't require sophisticated lab equipments. We are just using the RPA or the RPA isothermal amplification, and it don't require any other stuff, and can be performed in a single point of care setup. And BioScan is up to the WHO assured, like affordable, it's sensitive, specific, user-friendly, rapid, and robust. And we don't need any equipment. We just need simple equipments, and they can be delivered into the end user. And BioScan detection assay can be performed in less than 1 hour because the only amplification step is required, which normally take like 15 to 30 minutes. And then the running on the strips, it's just take like 2 to 3 minutes. And the system is working very well. And we just published this like last week on 7th of September. And the system can be used for any purpose or any specific detection of any specific sequence of any DNA or any RNA. So we have this format both available for the RNA or for the DNA, and it can be used in plants as well as even it can be used in animal genotyping and animal pathogens, including the human pathogens. And this is our BioScan team. We are working as a group for different like protein production and RNA production and plant sampling and all these things. I'm working with Dr. Magdi Mahfouz. He is our PI. And these like 10 and Ahmed, they are student. Shiva and Rashid, they are working as a postdoc in our group. And with this, I will say thanks for your attention. And if there is any question.
Thank you, Zahir Ali, for a nice presentation. So may I ask from audience if somebody have any questions? Can you please come to the stage if you have any specific questions, so you can ask from here. And Dr. Imran is coming.
Thank you very much, Dr. Zahir, for a very nice presentation. It's a really good system. I just wanted to know about the commercial potential of this assay because we are using biotinylated probe and usually they are expensive. So what is the cost of per test, and is it competitive with the already available tests?
The thing is like here, we are not using any biotinylated material. Like we are biotinylating our protein. So here, like as I explained here in the production, here, we are using biotinylated protein, so which can be made inside the bacteria.
All right.
So it's like, for example, we provide biotin to the bacteria, and we have this AV tag, which is responsible, and we have the BIR-A protein. So this BIR-A conjugate with the biotin, and it's bringing it to the AV tag present on the DCase9. So here we are not using basically the biotinylated material, and like if we produce this one, like one batch can produce the minimum for 10,000 samples. So this, we think that this is very competitive for agriculture use, because if we are using biotinylated, yes, as you are saying that if we are using biotinylated probes and all these things, they are really expensive and not feasible with agriculture system.
All right. Thank you very much.
So any other person, if you from the audience? Yes.
Okay.
Dr. Tahir, Allah Akshir, such a commendable job. Congratulations. I was just looking at GMO detection.
Yep.
And you said that you went for 35S promoter, right?
Yep, because this is a proof of concept experiment, so we use normally 35S, yes.
Well, did you try something from tDNA? By the way, that is tDNA, that stretch is mostly conserved. Even, even single promoter, double promoter, even something can vary in that part.
Yep, like this we discussed, and this was one of the reviewer comment as well. Like when we were submitting our paper, this was one of the reviewer comments as well. Yes, we can use the left TDNA border because it's mostly conserved and it's present as a, and this provide basically like, we just need 20 nucleotide sequence, and the TDNA left border, it's contain 24 nucleotide. So which is an exact target that yes, any integration through TDNA can be confirmed through this system. But we use the promoters because if the transgene is made through gene gun, then it can confirm it as well. So we can confirm any promoter, any conserved sequence, or any TDNA. Yes.
Right. Other thing, yes, you said that it's comparable to qPCR. That's really commendable. Have you just compared, and also have you compared its economic feasibility? I think the partly was asked by Dr. Imran as well.
Yep, the economic feasibility, like now we are using the commercial strips, which is like $3 a strip. And we are thinking that if we can make, like we are using these commercial strips. So if like, for example, looking into like the slide before this one, looking into the composition of the lateral flow strip. Yes, here. What these strips have, it's just like a paper, and then they put the streptavidin and the anti-fam antibody. And this is used, and then they are selling it like $3 a strip. So like our reagent, it's not more than $1. But the thing is like here, the problem is like this one. This is $3. So what we are thinking that now we are making our own strips because we can produce the streptavidin protein, and the anti-fam antibody is available commercially. So if like here we are taking 1,000 strip on $3,000, but on these 3,000, I think we can produce maybe 20,000 strips if we have assembled it by ourselves. So this we are thinking.
Thank you so much. Congratulations again.
Yep.
I think there are no more questions. So anybody else if you want to ask? No. So Zahir Ali, thank you very much for this nice presentation. So I think I think stop here. So thank you again.
Yep. Thank you.
So I think we go for the next presentation. Can you please turn this off? And, yes.
Next lecture is from Dr. Allah Baksh. He is scientist in Center of Excellence in Molecular Biology, Punjab University. He will give a talk on new frontiers in genome editing of plants and regulatory landscape worldwide.
Good morning, everyone. Before starting, I would like to congratulate comstech for putting up great effort. And especially I'm grateful to Dr. Emre and Dr. Imran for having me here. Thank you so much. I work here in this institute, National Center of Excellence in Molecular Biology. That is a kind of national asset. And it was developed back in 85, 86 under the Act of National Assembly. And why I'm saying it's a national asset, because it has produced a lot of molecular biologists. Now you can see them in the country. So we are based in Lahore. The mandate of the institute is to conduct research in different areas. We are right now talking about the plants. So confining to the plants, we work introducing different traits of insect resistance, herbicide resistance, virus resistance in different crop plants of economic importance. And like we produce a lot of transgenics. So whenever we develop transgenics, let me take my pointer here. Okay. So we produce, like I said, a lot of transgenics, a lot of transgenic events. So we need to confirm whether they're the risk assessment studies. The one particular lab They're working only for this sole job. And another lab there, they're working on edible vaccines. We are very good, I must say, at cotton, double and triple gene transgenic cotton varieties of this institute have been approved. They are already at farmer fields. According to Pakistan's national policy since 2010, we have transgenic cotton in field. So these transgenic cotton varieties developed by these institutes have covered approximately 20% area this year. So we are also working fiber improvement, physiological improvements, and one of the branch of the same groups of cluster of labs, they work with the commercialization of all biotechnological products developed at this plant section. So we use different tools, and whenever we talk about the tools, how we can forget genome editing. This is just a quick, like you can see, we started from the selective breeding to crossbreeding, hybridization. Then we started exposing our explants to source of gamma radiations by developing plants by mutagenesis. We got breakthroughs even in Pakistan, especially in cotton and pulses. Then we learned the art of inducing polyploidy, protoplasmian. In 90s, we saw the commercial transgenics in the field. And nowadays, since 2013, we got the genome editing. And this is the rocket that has been launched and is moving speedily. And at the same time, I would like to thank these super ladies. Because of them, I'm really talking about genome editing. So why it's trending nowadays? Genome editing is really efficient, cheaper. It's a big tool for a plant breeder. My basic training is in plant breeding, so I'll take it as a tool to introduce any trait of economic importance in croplands. So it's very directed, off-site mutations can be avoided, and this is the advantage of working with the plants. We can target any genes, and a lot of traits has already been introduced, and like you see, published paper every day. Why? Because why this technique is trending, but if you compare to plants developed by classical mutagenesis, you will find 10,000s of SNPs among them. Even if you compare two natural plants of the same from the same barley field of the same variety, still you will find SNPs. But this is the way you can introduce a mutation at a single point at a very predefined location. That's the beauty of this science. The people who are in plant breeding, they know very well when the traits are tightly linked with each other, it's very difficult to segregate them out in next progeny. This miracle can be done by genome editing. For example, this is the trait in tomato, trait for taste. You want to keep it in your plant. And the other one, this is susceptibility gene, let's say, to diseases. You want to get rid of it. So this is what, by conventional breeding, this is really hard. And this is the genome editing where you can exactly delete it out. A lot of advantages, a lot of applications in different sectors. You can edit your plants and you can develop insect resistance. For example, recently it was exhibited by Chinese researchers. They knocked out this CP71A1 gene that actually catalyzes tryptamine into hydroxylase. And by lowering the level of serotonin, assay level gets increases and the plant attains the resistance against insect pests. This is what they achieved in rice leafhopper. Also, by using this technique, you can produce modified plants where the insects, they will serve as a repellent. You can change the color. And on the other side, you can also edit the insect pests. Well, this is the talk of the town that insect pests have developed resistance, especially when we talk about pink bollworm nowadays. So we know that what has happened, the binding sites of the endotoxins, BD toxins have been mutated. So this is a CRISPR where you can exactly mutate those delete those mutated sites. And what will happen? Your plant will be more, your insect will be more susceptible to that Bt toxin. So on the other hand, we know the insects, they have got a very fast detoxification mechanisms. And what you can do, you can knock out those genes which are in, by the way, as a result of that detoxification mechanism, insects are clever. They excrete the xenobiotics from their bodies quickly. So you can knock out those genes involved in, for example, monooxygenases. And as a result of that, your insect will be again susceptible to toxins or in chemicals. Another very interesting concept is de novo domestication. You have your wild type, that is resistance against insect pests, resistance against diseases, but it's not a good productive one. Problem is with the yield. By introducing domestication traits, by multiplexing, what you can do, you can introduce, you can develop the plant with that particular trait, whether it's insect resistance or disease resistance, with good yield. And on the other hand, if you have a good yielding variety, but it's susceptible to insect pests and diseases, again, using the same technique, you can introduce a trait that will make it high yielding cultivar and resistance to insect pest and diseases. So there are a lot of market-oriented applications. So many things are coming nowadays. Every day there are papers, and mainly they're related to agronomic values, food and feed quality, biotic in abiotic stress, and herbicide as well. Yes, the gene rocket has really been launched, like it said, and it's moving speedily. This is a bit old data. In 2020, if you go to AMI regulated database, you will see that 86, and now it must be beyond 100. The cases have been filed that contains genome edited in plants and animals. What's happening in Pakistan? You have heard presentation from NIPG that were related to genome editing. Besides NIPG, the other institutes, they are also, for example, CAM and FC, they got a good heavy project. I must say the government bodies, they are encouraging the projects containing genome editing. Also, there is a huge project going on in Faisalabad, in different parts of the world, parts of Pakistan, projects on genome editing are going on. So this is a featured example from CAM. We recently did a project, PhD thesis, and it was related to potato. Potato is really important. Pakistan is one of the important producer of potato, and whenever we talk about the food security, the potato always occupies a special place because of its nutritional components, and also it's a hidden treasure. And in a country like Pakistan, we can grow potato 3 times in a year. So that's why it's very important. So, but the problem with potato, before that, we developed a very good genetic transformation system in potato using oripotrogenes, using internodal and leaf as an explants, internodes. And like you can see, it's not a difficult crop like cotton and any other recalcitrant crop, a big friendly crop to genetic transformation. So we have adopted this protocol in lab, and we are continuing with this for our next genome editing work. Are you all of aware with the coal-induced sweetening? So what we do, we store potatoes at low temperatures to avoid it's a sprouting and to avoid from the diseases. But what happens at low temperature, vacuolar invertase, they get activated. I mean, they break down the sucrose into fructose and glucose. And these reducing sugars keep on accumulating in tuber cells. And what happens, you get chips like these. And another disadvantage that these reducing sugars, they react with free asparagine, and as a result, there is a development of acrylamide, and acrylamide is a potential carcinogenic. So what we did, we tried to, at CAM, to knock down vacuolar invertase, and we developed constructs that were developed in Holger Puchta lab in Germany, and the constructs were confirmed as the routine lab processes. And the transgenes were generated, like I said, and they were confirmed by PCR and also by Sanger sequencing, by amplicon sequencing, and we found in one case, we found 1.03% modified reads, while in another case, 2% modified reads were noticed, were recorded. And with phenotypic analysis, if you see that the vacuolar invertase, there was a reduction in transcripts levels when analyzed by qPCR. And with the other target, again, the situation was the same. The transcripts levels were reduced. We also tested the Cas9 expression in different plant parts. And you can see, interestingly, that its expression by ELISA, its expression was high in tubers. Again, it was also confirmed by qPCR. The expression of Cas9. Quality assessment, again, the samples were stored and were sent to PCSIR for analysis. And PCSIR, by the way, is the complex of laboratories by Science and Technology, monitored by Ministry of Science and Technology. They're working great. And compared to, you can see the control that the percentage of reducing sugars was recorded less in genome edited lines. And again, to compare more, to analyze more, again, the vacuolar invertase activity was also calculated in these genome edited lines, and the activity level was less. And these, you can see from here, compared to the control, there's less browsing and fries. Again, the genome edited lines, when we compared them physiologically, we could not find any significant differences. So now I'll quickly go through what is, whenever we develop a product, what will happen with this product, how the regulatory authorities will think about, will take that product. So we have another presentation. I think Dr. Adil will also talk about something relevant to that. So whenever we say genome editing, it's a ZFNs, TALENs, CRISPR. So what we end with the SDN1, SDN2, and SDN3. SDN1 is just a simple indel with no any template. But SDN2, there is a template in it, and there is a gene modification at one or more positions. And in SDN3, why are SDN2, 3, SDN1, 2, 3? Because the rest of the slides will talk about only about these different nucleases, deletions. So this is addition in SDN3. You can see we introduce small at the long stretch of DNA, and that would be considered as a GMO. Let's see how the different regulatory authorities in different countries are looking at these types of SDNs. South America, they're very positive at the moment. They, like GMOs, they have also taken genome edited crops. And according to this paper published, SDN1 and SDN2 mediated mutations are not considered as a new combination of the plant genome. USA, if USA does not regulate or have any plans to regulate plants that could otherwise have been developed through traditional breeding, as long as they are not plant pests are developed using plant pests. So they're, of course, all of us know they're very positive. Japanese will consider them, will not consider them as LMO if just their bases deletion inserted are replaced with the natural repair of DNA strands. But when there is absence of remnants of inserted nucleic acid is confirmed, then the LMO status is exempted. So it will depend the type of SDN. Recently, we have seen development from Japan, which I will mention in next slide, and you will see what kind of products are there in the field now. Australia, SDN1 is deregulated, and SDN2 and 3 are regulated now. Russia, currently, there is a ban, but Russia is really embracing these technologies. They have a federal program of 1.7 billion US dollar, and they want to produce 30 gene-edited crops and animals by 2027. So some gene-edited products will be exempt from the law, which they passed in 2016. And this is the law that prohibits cultivation of GM. Here is the problem. I don't say it's better to use word challenge rather problem. So according to EU, they will be regulated as a GMO. But these are all, there is a consortium, SAGE, with the name of SAGE, European Scientists for Sustainable Agriculture, through genome editing. They have developed a consortium of 150 different organizations, and they're pushing the EU to reconsider the decision of the verdict which came recently. And they are mainly emphasizing on the definitions of GMO, and they want to try to assert that the crops with the genome edited does not fall into that category. We can look at that legislation, by the way. This is from European perspective, this mutagenesis, where you expose your explants to gamma radiations mercilessly, and there are a lot of chromosomal aberrations, deletions, still that is safe. And also we cross to different, let's say, to different varieties of same species and interspecies. There is exchange of material, still that is safe. But genome editing, this is just a mutation at a predefined locus. So that is dangerous from EU perspective at the moment. Let's see what will happen next. This is just the green color, the countries with the green color, they are really positive. And they are not regulating them as GMOs. But recently, you can say, I don't have any information, unfortunately, from Pakistan, but it is, I heard from Dr. Shahid that they're going to discuss it case by case. And the red ones, these are all where they will be regulated as GMOs. And interesting decision came from New Zealand High Court. This is also a very recent decision. They will also consider these genome edited crops as GMOs. These are the plants, different plants, which have already got permission for field trials in different countries. I will not talk about US, China. Interestingly, in Japan, they have actually commercialized this genome edited tomato, and it contains up to 5 times the amount of the GABA, the gamma aminobutyric acid, and that is believed to aid relaxation and help lower blood pressure. And also very recently, there is a decision by Belgium. In Belgium, VIB, very famous institute, in collaboration with the Flanders Research Institute of Agriculture, they have 3 different maize trials against climate resilience and low lignin. And also one of the post-Brexit, UK is going good and embracing these technologies. And recently this Romstad Institute, they have got permission for the field trials of ultra low asparagine wheat. But I presented their work of vacuolar invertase, and that was performed by Dr. Anila. She recently completed her PhD. Dr. Kuhn, the constructs were developed in KIT in laboratory of Holger Gupta, and Dr. Kuhn, he got the Humboldt scholarship. And Dr. Neela, she regenerated those plants, tested them in controlled conditions, did all the phenotypic analysis. And I also would like to acknowledge my institute that is giving us a full support. If you're on Twitter, please follow us, and thank you so much. I'll be ready to take any questions.
Thank you for your nice presentation. Any question? Can you come here, please?
No.
Okay.
It's okay. Bring the mic, please.
First of all, Dr. AB, I would like to congratulate that you're doing a great job. Really, it's a main reason for the failure of transgenic crops, not only in Pakistan, but across the globe. The main responsible problem in the transgenic crop is the development of resistance against the insects. And now I think all transgenic crops, they are no more long protected against the insects. It's very fortunate that some scientists across the globe and even Pakistan, they are trying to understand the mechanism of the resistance, mutations, particularly in case of the Pactinophylla gossypiana, pink bollworms, the whole receptors has been modified and the resistance have been reported in the cadherin gene mutations, which the research is still undergoing. I want to ask you, okay, when these cryotoxins or some other toxins, like in yesterday talk, Dr. Zahid has identified HVT and plant lectins, are they doing some sort of detoxification mechanism studies or metabolic studies so that we should know how these toxins are metabolized or detoxified when entered into the body of the insects? Like in cryotoxins, when it ingested by the insects, it enters into the alimentary canal or food tract of the insects. It targeted the food pipe and it ruptures that particular food tracts. An insect dies due to starvations and hungers and stops feeding. That's the real mechanism of the action of this toxin. But I don't know in HVT, what Dr. Zahid yesterday pointed out, it is neurotoxins and it just captures the neurological network of the insects. I just want to know, because you people are involved in these studies in depth in different world-known institutions, why we are not taking some metabolic studies, and so that we can know the detoxification mechanism, we understand the reasons, what type of enzymes are involved in detoxifying these toxins. Can you please comment over this one? Thank you so much.
Okay, thank you so much. Partly you all already answered. By the way, we have to be very smart, clever, and we have to integrate different technologies. I will not focus only on BT toxin nowadays. I will go for different RNAi methods. I'll go for the CRISPR-Cas. So what I will do, I will try to integrate different techniques to get some advantage, let's say using any of this technology against your target pesticide. So we have to be very integrated in that, in your first part of question. The second, you talk about, BT, we know the receptors of cryotoxin, but I think this detoxification mechanism is before upstream to the binding to their target. So once the BT toxin gets detoxified, like you said, there are different, whether we apply chemical toxins or it's a BT toxin, okay, that is ingested. With the chemicals, first is the cuticle, that's the problem. Even once it's entered into the insect body, then we have all these monooxygenases that they keep on detoxifying. So before binding to the midgut, the linings, if the BT toxin gets detoxified and is excreted from the insect body, that is the problem. It won't be efficacious in that case. So I would again say we have to adopt different technologies, conventional and non-conventional, together to combat these pests.
Assalamualaikum. Dr. Alavash, that was a tremendous work and great achievements. So I have a question regarding regulatory concerns. So have Pakistan regulatory authorities defined SDN1, SDN2, and SDN3? And
I think maybe Dr. Adil can tell us better in next presentation, but right now what I discussed with Dr. Shahid, he said that, yes, what we are actually as a CAM, as a NIPG, we are giving them information that this is the type of research work which we are doing at laboratories. For lab commission, no problem. But the question will be when we will get about what will happen when we will apply for the permission of field trials. That is the thing where the legislation need to be worked. And like Dr. Shai said, that there will be decision by case to case. And it means that we will have to decide, we will have to inform the competent authorities, they will decide later on.
I guess the competent authorities here.
Yeah,
So
I'm also waiting.
Yeah, it's already delayed. I guess they should recognize that. So thank you so much.
Just adding one point, I would like to explain. National Biosafety Committee, which is working under the Ministry of Climate Change, they are actually responsible to, I mean, considering the case-to-case variation. So this is the regulatory platform.
Yeah.
Thank you.
I think there is no question. Thank you.
Okay.
So next lecture is from Dr. Muhammad Adil. He is from Ministry of Foreign Affairs, Pakistan. He will give a talk on an outlook on regulatory affairs in the field of agriculture.
Bismillahirrahmanirrahim. I hope I'm audible. Assalamu alaikum everyone. My name is Muhammad Adil. I'm both a scientist and diplomat by training. It's an interesting proposition. I would like to congratulate the organizers for putting this forum together. It's a youth forum on biotechnology, so I'll try to, in the scope of the presentation, also try to cover what opportunities for youth are there for biotech in Pakistan. The presentation I'm going to give is going to be my own experience working at the interface of science and diplomacy, not an institutional position. So that's the disclaimer to begin with. The competent authority for regulations related to biotech in Pakistan is the Ministry of Climate Change. So we'll get onto that. So I think the previous presentation has provided me a very good and important segue to talk about the regulations. Whatever technology and the product landscape that we develop is contingent and dependent on the regulatory framework that is in the pipeline. So if you look at the biotechnology pipeline, you've got in academia, you've got industry, and then you've got the policymakers landscape. So the policymakers and this interface is extremely important. No matter how good your science in the lab is, if there are no amenable or ameliorating regulations, it won't work. This is the spectrum of regulations that operate across the world. Any regulatory system related to ag biotech, the products and the services that are on offer, they can be divided into these 4 quadrants. So ranging from promotional, permissive, precautionary towards preventive. So in the previous presentation, you saw the world map. If you proceed towards North America and Latin America, they are more towards the promotional side. So they do provide their enterprise and industries full patent protection. There's speedy approval. There's no restriction on imports and no labeling requirement. And then you gradually proceed towards like what Australia and many of the countries in Asia are doing. It's a more permissive and a merger of precautionary. So there's a more talk about plant breeder rights. There's more talk about, implementation of standards and some level of labeling. While what Europe is doing is most towards the preventive side. So there's a lot of restriction on IPR. There's also this discussion on socio-economic consideration. So you can divide the continuum of biotech within these quadrants. So at the end, we are at the mercy of these triggers. Any regulatory classification system that operates within the market today can be classified into a product or a process based. Gene editing has disrupted this space massively because you can't just classify something into a process and a product anymore. With gene editing, you have no means of verifying if it's edited. So with STN1, so it raises a lot of questions on whether the regulatory system is fit for purpose. So, but the contestation is that we have to proceed beyond the GM binary. We have it's not the scientists that have created this binary; it's just a commercial understanding. So if we look at how regulatory perception is being, understood within North America, it's more toward the product side, while Europe, Australia, they are more process-based. Canada, it talks about novel traits. So it's a whole mosaic. But somehow we have kind of got stuck into this GM binary. But it's the battle of the triggers that we have to overcome. And I'll talk about how we can overcome it. But this is like the classification regimes in the classic, in the GM model of the supply chain of biotech products. We looked at product and process. So if we look at how the global regulatory landscape is, evolving, uh, Argentina took the lead within this space. It deregulated SDN1 very quickly. Then, basically they have got some level of harmonization. Harmonization is important for biotech trade. If the country that you are exporting or importing from, if they don't have the same regulatory standard, trade won't happen. That is the biggest hindering factor for, biotech enterprises in the developing world. So if you look at the green part of the map, that's where, there are exemptions, there are facilitations, and there's also this important thing of relevant information and good to know information. So some countries, they ask, exporters and developers too much information. So that's where, Europe comes in. Australia recently took the ambit of, deregulating SDN1. Within Asia, in many of the places, there are still ongoing discussions, but I think there is some level of consensus on exempting SDN1, because frankly, there is no way of verifying that. And it abides by the understanding of conventional breeding as well. So, but Europe remains the odd party out within this map. So if you look at, what is the need or what is the understanding? So I'll also talk about domestic versus international regulations. I think the international regulations, where international law, especially with regards to biological weapons convention, with regards to WHO comes in, it's also important. We as, practitioners of biotechnology and young biotechnologists need to be aware of these interfaces as well. So, one of the things that is that, this increase in regulatory requirement is not a result of scientific assessment. Scientists are doing their best by, publishing these results and being very transparent about the process. It's the peer review, the science as the way it's practiced, it's very honest in that sense. But there are social, legal, and political constraints. So that's the debate within the European Union as well. How do we include socioeconomic considerations? But then as scientists, how do you quantify socioeconomic considerations? That's an important debate. Then there's also this contradiction between international law on some level. So there's the Cartagena Protocol on biosafety, which discusses the regulations or transboundary movement of living modified organisms. But on the other hand, you have got sanitary and phytosanitary measures in the WTO, World Trade Organization regime. They've got different mandates, but there is still no consensus on how they define biotechnology. In some places in the Cartagena Protocol, gene-edited products are classified as synthetic biology. We know as scientists that is not correct. So this contradiction which needs to be addressed as well. And there's also this false dichotomy of nomenclature. We keep on saying that, and there's this need for us as scientists to, keep hammering this point that the understanding of GMOs is not a workable framework anymore. The product landscape, the tech landscape has evolved so much. There's a lot of convergence, synthetic biology, bioinformatics coming in, that we need to move beyond, the jargon of genetic modification. And then, if we set the regulatory standard, too high, it encourages monopoly. So one of the things that was talked about in the early stages of gene editing was that it has allowed democratization of, gene editing. So more startups were coming into the space, and it was providing, more opportunities for young startups. But that's something that we need to overcome. Institutional drift. So if you look at the graph of regulation versus technology, technology is always outpacing regulation. So that results into an institutional drift. And it is, this interface between science, diplomacy, and policymakers to reduce that institutional drift. So giving you an example of how countries that have deregulated SDN1 or have talked about, looking at differently gene editing approaches, they've looked at these 4 options. So many countries conducted technical reviews. This is from Australia. So they talked about, basically they do public consultations. So there's on the websites, they give a draft that, we are thinking about reconsidering regulations. So how do, different stakeholders perceive? So these are the 4 options that were on the foray. First was that no amendments to the, gene technology regulations, and different countries will have variation to the words, but it's still the same landscape for everyone. With option 2, regulate certain new technologies. For example, let's say we exempt SDN1 and then we add ODM, SDN2, SDN3 within the framework. Or, option 3, regulate some new tech based on the process used. Or option 4, exclude certain new technologies. So these are the options that are given to stakeholders to write on, that how do they perceive and justify their positions. So what were the key themes of that, and this review actually resulted into SDN1 being exempted. One of the important key themes was that stakeholders highlighted that gene editing is different from genetic modification. Inherently, it's different. The characteristics of the final product, not the process. And this is very important. If the product is, similar to, we talk about substantial equivalence. then there is no need to create an unnecessary, a new regulatory framework. Gene editing, by virtue of its cost structure and the business model, it can deliver a new green revolution. It can democratize agriculture biotechnology because it's cheaper and there's this less friction or barrier to entry. On the other side, the other side of the half, with the lobbies and NGOs, they talked about that gene editing should be regulated. There's a threat to organic industry. So it's, again, if you look at it, it's still the binary in the way the perception is being pursued, that they have got a pro versus an anti-lobby and harm to consumer choice. So these were the thematic outcomes, but it did resulted in Australia deregulating SDN1, which brings me a very good segue into international regulations. So we talk about domestic regulations, but what happens at the international foray? The way international law operates is that, when these treaties and conventions, such as, the Cartagena Protocol, then the Biological Weapons Convention and international health regulations, they do provide guidelines which are absorbed by domestic legislations, and we actually, update or change our laws based on these international regulations. So the idea of these regulations is to, clearly classify what are the rights and obligations of countries, how they can, reduce uncertainty, facilitate trade between different countries, prevent conflict. So these are the broad ambits of international regulations. So that's what, diplomats are supposed to exercise, sit in these multilateral institutions and debate and negotiate on these issues. So if you look at the international regulatory landscape of biotech, in some places, like I've heard that biotech is not regulated on the international. I think it's heavily regulated at the international level. So you've got a whole spectrum ranging on the end use of the biotech supply chain. So on one side, you've got arms control. So the Biological Weapons Convention talks about biosecurity, biorisk. And these are good intervention places for young biotechnologists, because these interfaces, especially these conventions, their secretariat offer a lot of engagement for young biotechnologists. So if you simply Google any of them, these conventions, they've got dedicated secretariats, and they do offer a lot of engagement activity on youth advocacy and how we can be mobilized to provide, good policy and regulatory advice. Then if you look at the second side, it's the health and disease. So we are talking about international health regulation with regards to WHO, the Plant Protection Convention. So they are all impinging some in one way or the other on how biotech should be regulated. Interestingly, there's no consensus on how they define different suites and the product landscapes of biotechnology. Then you've got environmental protection. You have got the convention, Cartagena Protocol. It talks about if living modified organ. You can see there's a difference in jargon as well. We say GMOs, there's LMOs, and there's whole differentiation in semantics. But at the end, semantics are important. Then if you look at trade, the World Trade Organization, then social impact. So it's a whole spectrum of international treaties that are impacting the way we practice biotechnology. So one thing you practice in the lab, but it has a higher umbrella of international regulations. So what are the key issues that we need to be writing on, discussing on, deliberating on? First, importantly, is institutional drift. Tech will always evolve faster than regulations. So how do we reduce it? By writing more and more about risk safety, how we are developing voluntary guidelines and creating risk structures that are proportionate to technology development. Try to reduce treaty conflict by more discussion. All of these treaties, they've got science advice mechanisms. So it is an opportunity for scientists to actually work within these treaties. They've got within the multilateral system, there's scientific advisory board. The Cartagena Protocol has an ad hoc technical expert group. That's places where you can go work and give tangible science advice on. So we need to debate on whether these treaties are discussing relevant things, or if there's a clash, how do we get over the line? Then defining risk. We do talk a lot about interdisciplinary research, but unfortunately in biotech, I haven't seen this interface evolving too much. People from natural sciences don't mingle much with social sciences. And even if they are, they're not writing on these issues on how do we define risk, especially with the dimension of science, with socioeconomic. And there's also this infodemic that, a lot of propaganda, you go on, there's a very important study recently that talked about the impact of fake news on commercialization of biotech. So the survey showed that more than 60 to 70% of the people actually use Google to search what are the implications of biotech. And the first hits they keep on getting are propaganda news, alternate websites talking about how we are creating these aliens and Frankenstein monsters and stuff. So there's a need to overcome these barriers by becoming more and more transparent and open about the processes that we do. Crucially, convergence. And this is where also the other side of the interdisciplinary education comes in. So on nano, quantum, and bioinformatics, there are no international treaties. It's an unregulated space. But as new and new products come into the landscape, there are going to be discussions on regulations. So we have to capture on these, areas right now, that how we can proceed forward with regulations on nano, bio, quantum, bioinformatics. So convergence also remains a challenge. So this gives me a problem, like a segue towards what science diplomacy entails. So now that we have discussed regulations, we can see that any of the problems that we are trying to solve through biotech, it can be resolved into resource and societal needs, so food security, climate change, health security. So they all have a scientific dimension. The problems that biotech is trying to solve, they are cross-border and global in nature. A single cluster, sitting in Boston cannot solve the world's food hunger. It has to be a collaboration. So science is a global enterprise. Global problems have a scientific component, and the way we practice science, scientific method, is one of the best available tools for solving societal challenges. Because science, the way it's practiced in Pakistan and anywhere in the world, it's the same protocol. It has harmonizing value. So science diplomacy, it's a merger of these interfaces of science and policymakers, how they can come together, bridge these gaps, and solve these grand challenges. Because science-based knowledge, it keeps on evolving. So there is this value in connecting with the policymakers, with diplomats, and advising them on how it can be utilized. So it requires scientific and diplomatic literacy. So how do we bridge this with biotech, with gene editing? So one of the things is that there have been asynchronous regulations, and they've been largely due to unscientific reasons. But despite of that, there have been 20 years of history of safe usage. So if you look at our smartphones, if you look at AI, it's an uncharted space. But if we compare it with biotech, it's so heavily regulated, despite the fact that we have got history of safe usage. The European Union uses the precautionary principle. The precautionary principle states that if technology has a propensity of harm, it needs to be regulated. Again, no boundaries. So they can just regulate literally anything under the sun, under the precautionary principle. So science diplomacy, this interaction between policymakers and scientists, it can bridge the gap. It can enable to render science advice and also create these large scale infrastructure. These are very important. So you've got example like, CERN, you've got example like ITER. So these are large scale infrastructure. It is not possible for one country to have massive sequencing facilities. And so we need these more collaborations. For example, in case of ag biotech, we've got CGIAR, ERI. So many of these platforms are avenues of creating consensus between countries on regulations. We need to better utilize this. So three main deliverables that we can use. First is capacity building, and that's where the young aspect of the youth forum comes in. How we can design these activities between different countries, and that's where we can facilitate on providing capacity building in different areas. Scientists that want to engage with diplomats, how do we do it? How do we draft policy memos? Because there's also this, in my interaction, I've seen this tendency to use jargon. So, these policymakers, they don't have enough time. So how do you deliver the correct information in a limited amount of time? That is very important. Using the same regulatory language is very important. So we talk about synthetic biology, LMOs, GMOs. We need to use, a similar language. We say genome editing. We should start using gene editing, because at the end, it's still a micro level that we are changing. It's not the genome. So when you say genome editing, it has a perception in a person's mind that you are changing the entire genome. It's a small gene. And we need to talk about more coordination between treaty regimes. And that will happen when you have more interfaces between natural and social sciences. So how do we, and then that leads me towards consumer. So consumer perception is extremely critical if we want to take our crops to the market, because we have got within Pakistan, we've got an excellent academia. More than 50 biotech institutions working across the spectrum on any number of issues. But how do we get it to the market? So there's this need to reflect clear consumer benefit, the products that we are developing, the traits that we are working on, what is the consumer benefit that they could have? And then more liaison with industry. That holds true, especially for the developing world, how we can attract those investments. And we have to have constructive dialogues, this is what PABIC, like Pakistan Biotechnology Information Center, has been doing, to have these dialogues to promote this understanding. And lastly, facilitating market potential of deregulation. Like SDN1 has been deregulated. It means it is not like under a regulatory cover. So why are we not trading on it? So these are important market trends that we need to be aware of. So what is the future that we are looking forward to? So there is some level of regulatory harmonization in the case of gene editing. SDN1 has been deregulated in more than 50% of the world. So how we can benefit from it? That's the question for us to answer. But if there is no harmonization down the line, the crop industry will face the same challenges as it has faced in the case of GM. And in either way, the situation, the climate catastrophes that we are facing, there's a drought in Europe and wildfires in Australia, there's a more need to adopt GE technology. So we need to produce food fast and in a small piece of land. So, and the idea is that, and I cannot get into too much detail, but the definition of SDN 1, 2, and 3, it's still in a fluid spectrum. So we need to develop consensus fast on these terms. I've been in dialogues, like for example, in Philippines, they actually talk about number of bases. I think it's an obtuse understanding, but still we need to develop consensus on how we are going to proceed with SDN 1, 2, and 3. And lastly, science diplomacy and these international discussions involving youth, because the youth has the energy for advocacy as well. By conveying the challenges that they're facing and the kind of, the expertise that they have, it can enable GE regulations. And it can basically allow us to, benefit from these technologies. With this, I thank you for your time and happy to take questions. Thank you.
Yes, so please.
Thank you for your nice presentation. Any question?
Absolutely, like that's the heart of the issue. And there are interfaces. For example, like these treaties, Cartagena Protocol, the Biological Weapons Convention, they have on annual basis have a meeting of state parties, and then every 5 years they do a review conference. So scientists should be participating in these dialogues. I think it's important to have a seat on the table, scientists and policymakers sitting together, and it's perfectly possible. It's more about increasing opportunity. For example, like in the Foreign Office, I look after the Biological Weapons Convention. So there's an upcoming 9th review conference, which will discuss and deliberate on what is the future of biosecurity. So that's where scientists will get a lot of say. So those are platforms where we need to start, within our university and academia, start talking about how we can better, participate in these forums. And raise issues on what does academia finds uncomfortable with regards to the policy. Do they want to deregulate something? Do we want to commercialize something? That dialogue is still needs a bit of optimization on what our requirement is. We are very good at publishing and all of this spectrum, but conveying the commercial interest, it's just still somewhere in the middle. So we need more participation. Absolutely. So like, many countries like ISAAA, the International Services for the Acquisition of Agri Applications, does a specialized course on science communication. Science communication is an important domain. Like, science diplomacy is another area, talking about trade and commercialization to biotech students. So like, I did my bachelor's from FC College. So we did have a course on commercialization of biotech product. That changed my perception that, while many of us want to work in the lab, some of us want to work in the policy space. So that landed me in the Foreign Office. So there is this need that we need a good distribution of scientists that work in the lab and scientists that work in the policy. So, and you need more of these people in the right places, climate change, Foreign Office, and, commerce and trade, so that they can actually, be a voice for the scientists in these forums. And that will happen when universities have capacity for interdisciplinary dialogue. So people from economics can better guide biotechnologists on how they need to, draft proposals that convey the trade or the economic benefit of developing biotech traits. So that's something that should be added in the curriculum.
So any more question, please?
Thank you so much, Dr. Adil. I'm Dr. Favan from University of Agriculture, Faisalabad. So I have two questions. First one is regarding ethical concerns about SDN1 especially, because as you said that we are unable to detect, for example, which technique has been used to modify this gene. So what about ethical concerns regarding this one? What do you think about, for example, here ethical concerns in Pakistan, as well as in what?
I think like these are important questions when we talk about risk assessments. Yes. So if we talk about ethical oversights, any biotech lab that operates still has in the university, they do have an IBC, an institutional biosafety committee. Mm-hmm. So any research that is happening, it's still on paper. So even if it's edited by SDN1, they still have declared it that they have, that's, so first thing is voluntary assessment. And that's what been happening in, places like Australia, Canada, US, where SDN1 has been deregulated. Within Pakistan as well, we do have a structure of IBC. And at the end, you still have that information Australia is talking about, voluntary declaration, that if it has been modified, EU is talking about, even if it's, let's say, if somewhere down the line they do deregulate STN1, you still have to label it. So there's still debate and discussion going on that how we develop ethical oversight. And I think it's a lifelong process. The ethics on dual use research, they're always evolving. Same will hold true for STN1. So I think voluntary assessment is the way going forward.
Okay. My second question is, as you know, that the future of genome editing is not just to knock out or just insert one gene, but it's going to move towards multiplex genome editing, especially about genetic circuits, metabolic engineering, as well as DNA recording as well. So how do you see that, how that regulation will be moving regarding especially multiplex genome editing?
So that's an important debate from the direction of convergence. That's what I talked about when I talked about nano by the interface. So with regards to convergence, the jury is still out on it. Like, when we get to that stage, there's going to be a lot of discussion on what applies to this. So we'll probably see some repeat of what happened in the case of GM with the same multiplexing. But because STN1, it would have been mainstream so we are looking at somewhere down 5 years down the line. So we're probably looking at a kind of mixture of this. But it's important that when scientists, they start writing on these issues right now, that this is the product landscape that's going to come in 5-10 years. How should we, as different communities? So we have got epistemic communities. You've got scientists, you've got social scientists, you've got policymakers, legal people. So they need to write on how their positions. That's what countries like Australia are very good at. They very clearly state what scientists want from the industry, from the government on their expectations. So I will see like it's going to be a combination of what has happened with the with GM as well. So it's going to be a combination.
Thank you.
So so this will be last question, please.
Okay. Thank you so much. My name is Wasim Sajjad. I'm assistant professor, and I just have one case study as an example for what are the reasons that the new biotech startup or the foreign investor not coming to Pakistan. I work with one of the startup that is Stanton Solution, and they're working on the climate change and agriculture sector. But basically what happened, they're supposed to install the technology at Chenab Head Marala station, and we approached the regulator at the local level. So you asked to approach the provincial level, and after the provincial level, we are asked to approach the federal level. So I mean, just like for the adaptive approaches, like special technologies zone authority is created for the digital technology, and also Pakistan single window initiative for the digital and IT sector. But unfortunately, and even for pharmaceutical, we have a draft, but unfortunately for biotech sector, we don't have a single window of operation, not only to reduce the cost, but also the timeline. And the task was given just like for 42 days to set the regulatory affair, and our neighbor country like India and Bangladesh, they did it and they launched the project. But unfortunately in Pakistan, still we lack the initiatives, there will lack the intent. And when I approached a different regulator, there were like a very delayed and decision making. So if it is the mandate of the science diplomacy, so they can initiate like just a single window operation to facilitate such process. Thank you so much.
Yeah, it's a good intervention, and I'm very aware of your good work that you're doing and your team as well. With regards to science diplomacy, that's something that we raise. We need to have these more interfaces. We also inform stakeholders that what is happening in the world, what are the best practices. And that's something that we raised when the special technology zones was launched. That if you're talking about commercialization of biotech, are you aware of the number of NOCs that stakeholders have to take to get their product commercialized? So that's an issue that we have raised. So advocacy is important. And we are also trying to raise on the issue on having a clear position on gene editing. Because when we have these consultations with other countries, we always talk about the potential of collaboration on ag biotech. But when we are asked about the regulatory landscape, it's still in the gray area. So the advocacy part is, very much on. But I think there's also, there are some movements in time as well because of the ongoing situation with floods. I think it's a good opportunity in the way to, keep raising this issue. That now that we have a national catastrophe at hand, we need to be good at tech adaptation, and we have a tool at hand. Like our scientists, they have done excellent work with regards to publications, with trade development. We just need to commercialize these things more and more. So what I saw in the lab can actually get on my table. So the advocacy part is happening, and I think hopefully you will see some level of improvement in the coming time on that. But the point is very well taken. Thank you.
So I think
I think like, to be honest, there is a level of understanding and there is this reception as well. We just need to get our act together with regards to, like, we have got the 18th Amendment impacting on environment. So the subject of environment is devolved. So, but there's still this lack of medium. I think it's from a problem from both sides. Scientists need to, like, I've not seen any publication from within the biotech space that, publishes policy memos from our side. So we took a little initiative, like the Foreign Office has got its own publication now, Science Diplomacy Perspectives. So we've tried to raise this issue. So I think, like, not just publishing research papers, but also policy memos, and getting in touch with organizations like CGIAR, IRI, ISA, which is the International Services for Agri Applications. They do provide tremendous opportunities for raising these issues. So when you do have these interfaces, you can, there is absorption within the policymakers on these issues.
So I think we thank Dr. Muhammad Adil for a nice presentation. So clapping, please. There is one announcement from Dr. Amin. Actually, want a group photo outside as cameraman is available. So
yeah, group picture for all of you. There will be 3 flags, one flag of Turkey, Pakistan, Kazakhstan. The people who came from Kazakhstan are on the behalf of IOFS, they will be standing with the flag of Kazakhstan. The people who came from Turkey, they will be standing up with the flag of Turkey. And other participants will be distribution, some with Turkey, some with Kazakhstan, and some with Pakistani flag. So we are just going outside.
okay, so we will continue after the group photo. Sorry for delay. Our next speaker is Dr. Zajipa Spekova. She is researcher at Breeding and Biotechnology Institute of Plant Biology and Biotechnology, Almaty, Kazakhstan. She will present her talk on wheat bacterial diseases in Kazakhstan and identification of bacterial population. So with this introduction, I request Dr. Zed.
Assalamu alaikum. Firstly, I'd like to thank the organizers for invitation for this amazing forum. Also, I would like to thank Dr. Emre Aksoy from Middle East Technical University, Turkey, for informing about this forum. Also, thanks for Dr. Allah Bakish from Center of Excellence in Molecular Biology, Lahore. My topic on bacterial disease of wheat and the identification of bacterial population. Kazakhstan is the most important wheat producer in the world. We are top of 10 largest wheat producer in the world. We annually produce 14 million tons of wheat in average. In the half of production, we export to European countries, Middle East countries, and we have very good, high quality of wheat. Actually, we grow wheat, spring wheat in the north part of Kazakhstan. Oh, sorry. Yeah, you can see from that map. And in the east part, in the source part, we grow winter wheat. Bacterial disease, most important, 2 kind of bacterial disease. First one is black chaff or bacterial streak. Pathogen of bacterial streak is Xanthomonas translucens. Is it comfortable condition for black chalk is humid weather? And you can see the different symptoms on the spike and the stem and the leaves of all cereals. It can damage all cereals. This map presents us a global distribution of Xanthomonas translucens, potavar translucens. This is it. It has been conferred in 34 countries in the world, in African, European, US countries, and the Central Asia countries. Second bacterial disease is bacterial leaf blight. Pathogen of this disease is Pseudomonas syringae. It can destroy during flag leaf emergence of cereals, especially wheat. Also, you can see the distribution map of Pseudomonas syringae, potavar of syringae. It is a database of from KABI. You can see this disease distributed in all area of wheat growing areas. Also, this pathogen can destroy fruit crops, especially stone fruit crops. And another potavars of Pseudomonas syringae is Pseudomonas syringae atrophaciens. It's also most important bacterial disease of wheat and cereal or other cereals. Also, we also have in Kazakhstan this disease. Purpose of our project was ecological monitoring of bacterial disease all wheat growing region of Kazakhstan. We collect samples from different wheat growing part of Kazakhstan. From west part from active region, and from north part from Kostanay, from Nur Sultan, Karaganda, and the Pavlodar region, as well as east and the source part of Kazakhstan from Chimkent, Almaty, and the Taldyqorghan. We collect the samples to identify bacterial population. In these tables, you can see primers. We use lamp primers to identification of these species. Also, we use specific primers for IT's region of bacteria, also for Xanthomonas and Pseudomonas species. This table presents to us some results of monitoring of bacterial disease in Kazakhstan. We were observed in the Kızılırda region, Aktobe region, Kostanay region, Akmola region, Pavlodar, and Karaganda region. You can see the coordinates of this location, name of variety or name of crops. Also, name which bacterial disease we observed. For Xanthomonas species, we used for identification lamp analysis. And in this photo, you can see the control, and the second and the third probe was positive reaction of Xanthomonas species. We use because lamp analysis is more easy and more cheaper than conventional PCR. And you can use even for like short time. Also, this analysis doesn't need any thermal cycler in the different condition. Also, we use specific primers for identify pathogens. Firstly, we use for ITS region of bacteria 16S RNA Then we use specific primers to identify potavars and the species of Pseudomonas. You can see the results of electrophoregram, and yes, you can see the species of Xanthomonas. And according to NCBI, according to our sequence, we found Pseudomonas syringae, Potavar syringae, and the Pseudomonas syringae atrophatans, 100% homology according to NCBI database. Also, we found in Kazakhstan Xanthomonas translucens, Potavar translucens. In conclusion, we found using molecular analysis 3 potavars of bacterial disease. We used markers, specific markers for ITS region, also for LAMP analysis in the conventional PCR. And we found in the all wheat region bacterial disease, distribution of bacterial disease. And I would like to thank for my team. First person is General Director of our institute. He is a principal investigator of this program, and I'm principal investigator of this project. We have 7 projects on this program, and the first one of on bacterial disease is mine. And the second person is Malika Shamekova. She is head of our lab. Third person, Ruslan Kalendar from Nazarbayev University. He gave us consultation to identify bacterial disease. Also, he developed fast PCR analysis, which helped to us to develop test system to identification of bacterial disease. And the Dilara and Rahimbek, they are doing molecular analysis. And Gulnaz, she work on microbiological analysis. Also, this work was supported by grant, Minister of Education and the Science of the Republic of Kazakhstan. I also would like to tell about my lovely topic on rust diseases. During many years, I work on rust diseases. My PhD was on leaf rust, and we have a lot of experience. My colleague, I could also works on rust diseases. Everybody know about UG99, which found in Uganda in 1919. It is a very aggressive race of stem rust. This aggressive rust spread all everywhere, and we have observed, we was observed UG99 in the north part of Kazakhstan in 2016, and we lost 30% of our yield. Stem rust, we work on stem rust resistant genes. Nowadays, more than 60 estrogens have been described, and someone is ineffective to UG99, and someone is effective to UG99. Purpose of our research was identification of new donors of carriers of resistance genes to stem rust race UG99 and the creation of new variety of wheat. We also use marker breeding selection, and we use resistant parents and susceptible parents did crossing, and using molecular marker assisted selection, we selected advanced lines. Then we send our advanced lines different ecological conditions to test. For example, West Kazakhstan, East Kazakhstan. Also, we sent to test Kenya, natural infection of UG99, a hotspot of UG99. And we created new variety of winter wheat, such as Raminal, Ali Khan. Ali Khan, we gave a name of my son to this variety. And Dinara, it's results of my PhD work. Nazam. We sent 40 breeding lines to Njoro, Kenya, plant breeding station to test in natural condition of UG99. And we selected these 5 lines, which has shown moderate resistance and resistance to UG99. Using molecular analysis, we identified different SR genes in these lines. You can see different SR genes, SR38, SR28, SR39, and the other genes we identified in these lines. And you can see in this table, 5 lines was more resistant and had SR genes. Yeah, you can see they're all breeding lines which sent to Kenya for testing. Also, productivity of advanced lines is also important for us. We should compare to our local standard varieties. So in Kazakhstan, it is the standard variety Almalı, GTSU, Progress, and these 5 lines compared to the standard, they give good yield, good productivity. And they were carriers of resistant SR genes. In conclusion, we identified 5 advanced lines with high productivity and high resistance to stem rust, and these breeding lines will be useful for breeding programs, and they can be candidate of variety of wheat. And I would like to thank my institute. Also, I would like to thank for Kazakh Research Institute of Agriculture and Plant Growing, because all breeding process we do in this institute. And the experimental plots, also I would like to thank CIMIT in Turkey, because my supervisor Alex Margonov from CIMIT, and he helped us too much. Also, I would like to thank plant breeding station Njoro, Kenya, and you can see there are some articles linked to our articles. And I would like to tell about this program, because I spent 200% of time to investigate this program. It's program a little bit different from biotechnology, but we are trying to develop a new inspection system, certification system of seed potato and the planting materials of fruit crops. Before, we didn't have this kind of certification system. We work on old Soviet Union programs, and we would like to develop this system in Kazakhstan. This program ordered by Agricultural Minister of Kazakhstan, also from business structure, from growers. We have 5 objectives under this program. First one is develop inspection system of seed potato. Then we are developing for fruit crops for 4 crops: walnut, apple, raspberry, and the plum. Then we are conducting training for field inspection and the lab inspection of seed potato and the plant materials of fruit crops. And the fourth task is development of test system to identify the detection of virus or other pathogen diseases. So we are trying to develop 5 test systems. We already developed 3 test systems for these 3 crops. Also, we are created new database, new system, so all farmers can apply, can do parcel declaration, apply for inspection. Then our inspectors will receive the information about growers. Then they will do inspection and download the results of inspection to database, and all growers can see all the materials of inspection, and they can print certification themselves. And our inspection system will give labels, virus-free labels and free labels for Erwinia millivorophaya blight disease. Also, for implement this program, we developed, created new official body. It will be private, but will be controlled by Ministry of Agriculture. So what we will give this body will do, this official body will conduct variety testing, US testing, and the genetic passport of plant variety. Also can identify pathogens in accredited laboratory. Also we need minimum 2 accredited laboratories, and also we will have training courses for local inspectors. Also we'll give consultation for seed producers, and our inspectors will find the quarantine object, we will inform quarantine service of Kazakhstan. To investigate this program, we have a contract with Netherlands service inspection system, because before starting this program we did review all the inspection system of European countries, US. So we preferred and chose best system for us for transfer, Dutch system. They have 4 inspection system. One of is NAK, who do inspection on seed potato. Second one is Nachtenbau. They do inspection for fruit crops and the ornamental crops. This program actually do fruit and vegetable together with fruit and vegetable research institute in Kazakhstan. The results of this program, we will develop optimal model of inspection service in Kazakhstan for seed potato and the planting materials of fruit crops. And step by step, we will increase number of crops. We will increase add some cereal crops, and we need time for that, because Kazakhstan is very young country, and the Dutch system, they spent 85 years to develop this system. Expected results of this program, we can in our group we work with on legislation for seed potato and the plant materials. We use old standards of Soviet Union. For fruit crops, we don't have legislation. So we would like to work on European standard, but we will add some our requirements, because we have our own list of pests, pathogens. This program will give us to avoid corruption in the inspection system, and it will give opportunity to grow to welfare of our growers, for farmers, and we do a lot of organize a lot of seminars to explain for our farmers. It is benefit for them, because without support of farmers, we can't work. Also, we have a group in this program involved more than 30 persons, and one group work on legislation on the government level. Also, we have group IT specialists who develop the database and develop this program, and we have molecular biologist group who develop the test system. This year last year we selected 4 inspectors for fruit potato. It's my team, and the second team is on seed potato. Previous year we have online training from the Dutch side, from NAK and the Nachtenbau. And this year we have 3 trainings from Nachtenbau. Already we got training on malus, on apple field inspection, and also we had inspection on rubus, raspberry. And next week our lab specialist will go to Netherlands, to Nachtenbau, to learn routine and the development routine and the diagnostic lab, to study system, how can be organized this lab system. And the other group already got one time in on seed potato, and they will go on October to study post-harvesting inspection in the NAK. Also, I'd like again to thank for organizers and for our ambassador. Thank you for your attention.
Thank you for your nice presentation. Actually, we can skip question. We can discuss it in the panel.
Okay, thank you.
So next lecture is from Dr. Umar Farooq from Department of Plant Sciences, Chair Group of Bioinformatics. He will talk on next section in plants from bioinformatics perspectives.
Hello and Assalamu alaikum everyone. First of all, I would like to thank the organizers, especially Dr. Imran, for inviting me here and allowing me to present my recent research work, which is about the application of artificial intelligence for crop improvement. And I'm going to present a recent research work about my that was done in my PhD from Wageningen University. And also I'm representing NIPG Pakistan because I'm also employed there. So before going on, I would like to endorse the initial comments by Dr. Asim the day before yesterday, that there is a very little contribution by AI-based solutions for crop sciences in general, around 3%. So with this aim, I tried to put my little contribution into this area with the hope of having a larger impact. And the topic that I have selected, I'm going to talk about is the informatics perspective for genomic selection, which is an important tool for crop breeding. And if you can forget about the GMOs and any regulations, then this is the talk to be listened about. So first, starting with the introduction, I will like to talk about some issues and challenges that we can face during the AI-based modeling for genomic selection. And then I will be talking about some improvement strategies that we can employ for AI-based modeling. And in the methodological framework, I've been working with linear mixed models, machine learning, and the deep learning, which is quite popular these days, especially for imaging applications in crop sciences. So the genomic selection in theory is a new approach of marker selection. And it was initially proposed by Mewison in 2001. Where they proposed a methodological framework for utilizing the whole genome SNP markers. And they were aimed at predicting the total contribution of all the alleles, all the genes, or the SNPs in a genome for predicting a trait rather than phenotyping it. They were aimed at predicting the total genetic contribution in terms of allelic effects. So in that paper, they have actually laid out the foundation for genomic selection methodological work. And later on, people tried to explore it and apply it in different breeding systems. And they were able to get real practical success in terms of genetic gain. So by using the whole genomic markers, we are basically adding up more information to the models. So it means that your selection accuracy can be improved and increased with respect to if we compare it with the benchmark phenotypic or pedigree-based selection. So this means that the genetic accuracy, this term in this equation, can be increased. And this is a numerator. So the other term is the genetic variance, which can also be increased by incorporating the genetic markers. And if we have a higher, DNA level information, then the total genetic contribution calculated by the genetic markers, it is fairly higher than the total genetic variation that we can calculate from the phenotypes or only the pedigree. So, and also we know that in an earlier presentation by Dr. Mansoor, he has mentioned that the genotyping cost is, with respect to the Moore's law, is at minimal level right now. So we can genotype a whole genome for around 50,000 markers in about $20. So it means that we can genotype a larger population with minimal cost. So the selection intensity will also be increased. So all the numerator terms are actually getting benefit for this framework. And since we are able to pseudo phenotype the individuals rather than actually phenotyping them in the field from the genetics only, from well before than the actual phenotyping. So it means that the denominator will also increase the impact of final genetic gain over time. So overall, all of these contributors actually try to improve the genetic gain over time from the conventional breeding process. So how it goes? So first we need to have a reference population, which is the most costly business of this technology. We need to have a quite diverse and having a larger effective population. We need both genotypes, usually using high throughput systems, and phenotypes. It's better to have high throughput phenotyping systems to minimize the system noises. And then we develop a model, which is lying in the middle. Sorry. Here. So that model is basically can be developed from any statistical or artificial intelligence framework that utilizes both of these information. And then for the test population or the breeding population, we actually predict the pseudophenotypes, or I would say the total genetic contribution of those alleles, which is also known as genomic estimated breeding values. And then based on these GBVs, we rank the individuals to select for the next generation. So what are the issues and challenges for this framework? So in this recent paper, I have categorized them into 4 categories. The prediction accuracy, you can see, is a compound function of different technological aspects, genetic architecture, population dynamics and statistical framework. So for example, if we have a very good technology to measure a large or millions of markers for a particular population, then there are still issues that you will be ending with multicollinearity and high dimensionality, which is good in terms of data. You are generating more data, but bad in terms of modeling, because you will be getting multicollinearity and redundant information for the from the modeling perspective. On the other hand, if you have a very low density genotyping, then it means the LD or linkage disequilibrium between the SNPs and the actual QTNs will be decreasing. It means you will be risking of missing some important QTLs from your population. And the other player is genetic architecture. Of course, it plays the key role. Like if you have a quite simple trait like monogenic or oligogenic, then and also the effect size distribution and their distribution over the genome, that is governed by the heritability models. And the overall heritability is also playing a major role into the prediction accuracies of the models. And the third thing, which is basically inducing a negative bias in terms of confoundings in the estimations. For example, the population structure is usually confounding the predictions. So when you are developing the models, you are basically risking of overfitting the models, and the estimates are usually not correct. So when you apply that in the breeding population, the realized and expected accuracies are quite different. So it means that this is a major player. I will show you some results about this as well. And then last but not the least, the statistical framework, which I am going to discuss in more detail today, which is basically also governing the prediction accuracies for a particular breeding program. So how to model from the very basic steps, I will give some basic information. So for example, if we have n genotypes and we have few QTLs or QTNs in this term, and then we have genotyped the population with almost homogeneously distributed SNPs. And we know that the phenotypic variance can be divided into genetics, environment, and their interaction. And with this modeling perspective, we are aiming at capturing the genetic contribution, which can further be divided into additive, dominance, and interaction parts. Whereas the Muvison's work was only basically centered at the calculating the additive part, but now with the advent of AI-based solutions, we have more flexibility to capture the other parts as well. So this set of biological problems can be translated into mathematical form like this, and you can see that this matrix, which is having many columns and a few rows, because in the rows you have only the samples or the reference population size, which is usually in hundreds or maximum in thousands, but the genetic markers are usually in millions. So what to do? This is not solvable by ordinary least square method. So you need to have some tricks. So for that, what you can do, so this is technically called the curse of the dimensionality issue, or phi is greater than n problem. The solution is to use some statistical tricks like shrinkage and regularization, and some other transformation methods are also available from the data science toolbox, or the feature reduction or selection is also a candidate approach for this. So in general, you can say that if we broadly divide the statistical or methodological framework into two parts, you can use the statistical learning method framework to predict the GBVs, but it requires the prior assumption, which is in terms of statistical assumption. So it means that if you are modeling a biological system, that biological assumptions should be closely matching with the statistical assumption. And this is the major limitation, because the traits have are quite diverse, and there could be plethora of distributions of traits. So it means that you can't make a separate statistical distribution for each trait. So it means that there is a trade-off and simplification step going on here. That's why you are the traditional or conventional methods usually lag with the lagging in the accuracies. And the other framework which is quite popular these days is the machine learning and deep learning stuff. where you do not have any assumption about your data and you just use your data as much as data that you can generate, and then based on that data you actually predict the patterns or the predictions. But there is issue with this approach, that you need a lot of data but you don't have any data because you have only few hundred samples, and with few hundred samples it means that and with high dimensionality issue you can risk of overfitting only on the training population. So yeah, some of you may be thinking that I'm mostly talking about the negative sides of having more data, but yeah, you can solve this problem with more data, but in an intelligent way. In a rational way, you can use an additional data to solve the problems owned by the high dimensional or big data. For example, you can extend your models using additional omics. Here you see we know that we can generate multiple omics data, and you can use that data to improve your predictions. And you can also nowadays generate the remote sensing data, like NDVIs, vegetative indexes can easily be generated from remote sensing tools like drones. So here the secondary traits or the simpler or component traits can also be used to improve the predictions. The third thing is the simplification, which I am going to talk about in the next slides, where you can use the dimensionality reduction and the regularization stuff. Here, the regularization can be statistical or from the prior knowledge. Like I was talking that you can solve this issue by having more data in a rational way. So you can use the prior knowledge, which is already available in the databases, in the free available sources. So you can use that data for your particular population. And the advantage is that your modeling will be more generalized and it will not be restricted only for a particular breeding population. So for that, I'll be talking about two approaches that we explored. One is prioritization, where you can actually incorporate the functional groups of these genes and SNPs, like gene ontology and co-expression clusters of using RNA-seq experiments. And then based on these, you can prioritize some parts of the genome or the SNP matrix to actually solve the problem with improved prediction accuracy. And the other strategy that I'll be talking about is restricted learning. I mean, you can, if you have a graph-based learning like deep learning stuff, then you can use the interaction information for genes and their products or the SNPs to regularize your model with sparsity. So you see that some of the connections can be totally omitted, and then the model could be very simpler for the better predictions. And so the first step that we did using linear mixed models and using the prioritization approach, it was basically using the Phenovator system, which is available in WAF engine. And we measured two traits. One is photosynthetic light use efficiency for 18 measurements. And we induced some light stress as well here. So in total, there was 6 measurements before the light stress induction. So there is an acclimation response here. And this is the projected leaf area, which was measured again by the reflectance of NIR imaging. And then we actually use the prioritization here. You see that some of the QTLs that we know that could be potentially the causal genes, or they could be closer to the causal QTLs. So we actually prioritize them in the equations by splitting the equation into two parts. Here you see that this group variance is basically the functional group of SNPs, and the rest of the variance is the same, just like before you have seen the equation. And then we observed that some of the Go terms and co-express clusters are actually significantly improving from the benchmark. Here you see that for the green is basically the PLA trait, and the other one is before and after stress induction for photosynthetic light use efficiency trait. And here you see that there is significant improvement from the benchmark for the knowledge-based models. But when it helps, this is a question. You can see that the knowledge-based models are actually more effective at the beginning of the growth stages for both gene ontology and coax knowledge. And for the later stages, when the heritability is quite higher, they are quite similar to the benchmarks. So it means that we can reliably or consistently use these knowledge-based models for improving the linear mixed models. And then we actually tried to cluster these clusters from either Go and or coax clusters into their semantic meanings. And we have observed that despite having many biological processes, there is only a few Go terms which are related to photosynthetic machinery. So we can see that these are clustered into a few clusters, like photosynthetic machinery is helpful to improve the predictions for those photosynthetic traits. And similarly for PLA, we were also able to get the photosynthetic machinery to improve the predictions. So now, I'm sorry. So now there are other research questions that which method we should use and when it should be used. And is there any clear opportunity for machine learning as well? So for that, we run some simulations, and we did some simulations with polygenic traits with and without large effect QTN simulated, and for both linear and nonlinear phenotypes. You can see that when we have very limited number of QTN simulated, then the effect sizes are quite higher. And when we have increasing number of QTNs, then the trait complexity or the effect sizes are decreasing. And similarly, for when we are using one large effect QTN, then that QTN has the potential to be easily detected by the model. So this is the scenario for the oligogenic traits. And these are the results. Here you can see that the machine learning and especially the ensemble methods have a clear advantage over the traditional blobs. And you can see for simple traits, they are quite similar to the Bayesian methods, but for the complex trait, they have still a clear advantage than all methods. And we tested this with the real data from Australian wheat germplasm for different traits. And we saw that for the cases when we have expected trait linearity to be lower, then machine learning was better. So for the confounding factors, we tested this using another data set, which is for sodium accumulation trait, where we have a large effect QTN identified. So it means that risk of missing that QTN is minimal. So we measured the impact of confounding factors like population structure. We saw that all the methods are sensitive to population structure. So we need to take care of this structure before. And the last part is about the deep learning stuff that we have done. Here, this is about, this is a fully connected feed-forward machine learning or the multi-layer perceptron model. You can see that if we have only 7 SNPs in the first hidden layer and the input layer, there is in between fully connected layers. So for each SNP, there is a connection from SNP to every neuron in the first hidden layer neuron. So this is quite time consuming in terms of training, as well as there are a blast of parameters, because for example, if you see that only 7 SNPs are here and there are only 4 in the first layer, there would be a multiplication in terms of number of parameters. So there will be 28. But what if we have 1 million SNPs? Then for only 10 neurons, very simple model in the first hidden layer, then there would be 10 million parameters at least. So this has to be solved. Otherwise, this is not realistic in terms of time computation as well as from practical considerations. So what we come up with to use the prior knowledge for this, we basically splitted this multiplication into subparts. Like using the prior knowledge, we actually divided this set of SNPs into subcomponents or groups. Like here, these from the interaction information of the genes of these SNPs, we know that these two SNPs and these three SNPs are actually interacting with each other. So why not to group them together and leave the rest of the connections? So what we did here is to map them to one neuron and map them these three into two. And why these 1 and 2? Because we took the cluster size of string interactions into place here. For example, if we have a larger subcluster of string interactions, then we actually weighted them more. And we saw that the performance goes up when we have more weightage to the more informative markers. So similarly, for the rest of the markers, we can map them to a single neuron and then so on for the rest of the model. But yeah, good thing is that you can see that this multiplication operator is broken down into subparts. Like you have only less than half of the number of parameters here left. So it means that the overfitting or the high dimensionality issue can be solved using deep learning and with using prior knowledge as well. So we run this approach on a simulated data with narrow sense heritability of 0.4 and epistatic heritability of 0.4 and with QTNs 10. And you can see that we simulated with 2 clusters. And then we observed that the prediction was significantly higher than the benchmark for the simulated data. And also we tested it for the sodium accumulation level trait for this data set. And then we saw that this CMI our model is quite higher in terms of predictions than the benchmark. So in a general conclusion, I can say that we can use prior knowledge, and these prior knowledge driven models can be used to improve the genomic prediction for complex traits as well. And we can say that the machine learning has sufficient room for genomic prediction. And we can also say that deep learning could be a choice for genomic prediction utilizing specific knowledge. And if you want to read the further stuff, then you can go through these papers. And I would like to thank the Wageningen team, Professor Dikti Ritter, who is chair of the bioinformatics group, and Aljan and Ham Navin, who are also machine learning and deep learning guys working over there. Thank you.
We thank Dr. Farooq for a nice presentation. So there are 2 announcements. Soon after this, we will start the panel discussion and answer session. But in the meantime, you can collect your certificates from the reception desk. And about 1 o'clock, we will have a lunch. Okay, just 2 questions. If someone asks to Farooq and Dr. Zajipa, if any specific questions, so just 1 or 2 questions, please.
So this is a complex question, and the answer should be expected to be complex as well, because there is not a thumb rule. We need more and more SNPs to cover all the trait associated SNPs. But yeah, how to know that? We need to incorporate some prior knowledge about that. Like, for example, if we know that this particular trait is governed by a particular QTLs which are located on chromosome 1, we can use the chromosome level genotyping only. So I mean, this is how we can reduce the number of SNPs using prior information. But yeah, we can use some feature reduction or dimensional reduction techniques, and we can do some pre-processing to reduce the noise for the modeling process. So I mean, this is a conjective approach. We have to use the integration of prior knowledge. We have to use the statistical knowledge to reduce the number of SNPs and markers to lessen the burden on the model.
So any question to Dr. Zajipa, then we will close because time is running out. So just we will wind up just now. Any question to Dr. Zajipa? So I think we just conclude this session, and in this session there were 5 presentations covering a lot of diverse areas, including genome editing and regulatory issues and other things. So I must thank all of the participants and the speakers. So Dr. Fahim, if you want to ask something, please.
Thank you for everyone for wonderful presentation. So,
ladies and gentlemen, thank you very much for your participation in our event for all these 3 days. With this, we come to end of our event. And on behalf of Comstech, I would like to thank our speakers and technical focal persons for all the hard work that they put into this event. I would also like to convey my special gratitude to Dr. Imran Amin and Dr. Emre Aksoy for making this, for doing a lot of hard work during the time of planning and execution of this event. With this, I request Professor Dr. Khurshid Hasnain, advisor Comstech, for his vote of thanks.
Thank you very much, Ms. Fazima, for giving me this opportunity to speak on behalf of the Coordinator General, who is not here, unfortunately. So we've come to the end of this exciting 3-day Pakistan, Kazakhstan, Türkiye Youth Forum on Biotechnology. And I'm sure it must have been a very learning and enlightening experience for the young people who were the focus of this activity. So let me just say that on behalf of Comstech, that we recognize biotechnology as a frontiers technology, one of those areas which are proving to be crucial and critical in a multifarious ways, not only in agriculture or plant or food security, but also, of course, in the area of medicine and health. And it is, of course, another example of an area where the different branches of science and technology come together, as in this case, the engineering aspects and the artificial intelligence aspects combine with the knowledge of life sciences to produce the amazing results of biotechnology. And so Comstech has been active in various ways in promoting biotechnology as other areas. As you know, our mandate is to create a cooperation and development through cooperation between the member states and their scientists. Our mandate is to promote capacity building, to enable the stronger countries to uplift, to help the growth of the potential of the other lesser developed countries. So we hope that activities like this, where we have been having exhibitions, where we having trainings, and we have having seminars, are a way of developing the capacity. And so, of course, we will continue similar efforts. Let me just summarize what I've learned about the participation in this activity. We had the participation of 8 countries, different countries in this event, with 22 international participants and over 100 local participants in physical attendance in the 3 different sessions. We had presentations from 18 faculty members and researchers, and of course, they have special importance for us because of the efforts that, of course, are required to make these valuable, enlightening presentations. We need to acknowledge the support of the organizations that have helped and supported Comstech in organizing this event. First of all, the IOFS, the Islamic Organization for Food Security. We thank them gratefully for their financial support. The ICYF, who provided us support, the International Islamic Chamber for Youth Forum, and the Pakistan Biotechnology Information Center, PABIC. All of them are gratefully acknowledged for their support to this event. And finally, we wish to thank all the participants, especially those who have come from abroad, who found time. And of course, we know that travel often is quite prolonged these days because of the unavailability of direct flights from various countries. So thank you very much for having taken all the trouble to come here, and of course, the trouble that will be taken to go back to your countries. We hope that we have been able to provide you with the good hospitality and to meet all your needs and expectations. And we hope that you had some glimpse of our country in this state. And some of you would be probably making the visit this afternoon. And I would be able to see some more of the physical beauty of our country. Finally, since Ms. Khazima would not thank herself, so it is my pleasure to acknowledge the hard work of the comstech team, who in this case led by Ms. Khazima Muazzam and Ms. Maham Jameen, who have done excellent work, I have to say. Let's give them a big hand. I also wish to acknowledge the administrative, the financial, logistics, and the services staff of comstech who have worked throughout these 3 days and even before very diligently, very efficiently, and thank you to all of our staff. So finally, have a safe journey to all of you who are coming from other countries as well as from other parts of Pakistan. And we hope that you will be able to continue, not just to have this one-off event, but you will be able to maintain your contacts and to continue your interactions and your associations and build upon this activity to further your research and your knowledge. Thank you very much for this patient hearing, and Assalamu alaikum and goodbye to all of you.
So thank you very much for your very kind words. And of course, I would like to thank all the comstech staff who has been of a phenomenal help for us during this event. With this, I request Professor Dr. Rashid Hasnan to give out certificates of appreciation to our resource persons. So first of all, I would invite Mr. Sami Sardar for his certificate. Thank you. Mr. Sami, just one more request, probably the last one for this event. Please give out certificates to other speakers along with Dr. Rashid. So I request Dr. Imran Amin to receive his certificate. I request Dr. Emre for his certificate, please. Guys, please cheer them up. Dr. Fahim Baloch. He's not here. Okay, so what's next? Dr. Sachkin. Dr. Suga. Mr. Abdullah Manafi. Ismail. We will request Mr. Abdullah Manafi to also receive a certificate of Dr. Ismail on his behalf. Dr. Asma Rahman. Dr. Imran, kindly receive Dr. Asma Rahman's certificate on her behalf. Dr. Zahid Mukhtar. He's not here. Okay. Dr. Makanob. Professor Dr. Makanob. Dr. Asim. Yeah, it's Makanob. How many? Okay. Dr. Makanob. Professor Dr. Makanob. Already. Dr. Shaheen Aftab. Dr. Laila. Dr. Nasser Said. Dr. Zaitipa. Last, Dr. Asma Imran, please. So thank you very much. Thank you, Professor Dr. Khurshid and Mr. Sami and Abdullah Manafi. With this, we come to end of our event. Thank you very much to all of you for joining us. The lunch is served outside. All the participants can get their certificates from Ms. Maham from the registration desk. Thank you very much.